Learners, this is Professor Salm, and I'm going to do a screencast on classification of prokaryotes, so that would be bacterial classification, and then we will break it down into chapters 19 and 20, gram-positive and gram-negative pathogens. Now this is material we've been covering for the past 3 weeks, so let me just point out before we get started with this PowerPoint, I just want to look at the content schedule for the course. So we're looking at the 14th today, or tomorrow, I'm sorry, is going to be the beginning of exam 2, which will stay available from tomorrow at 8 a.m. until Sunday night, the 17th, St. Patrick's Day, until 11:59 p.m. The students who should have posted their current event articles are the students with last name Krug, Nunez, and Parath, and also Richter, Reming, and Romaine sometime within this next week. And then all students are responsible for responding 2 times, or to either 2 different students or twice under the same article if there's one that's, keenly of interest. Next week we have spring break, no class, so no class this week or next week. You have, however, been assigned a fungal pathogen in which you will be doing a on an individual basis. Each student has been assigned a fungal pathogen to do a quick reference brochure in much the same format as our bacterial, like the 2A and 2B brochures that you've done in groups. So that is something that you can work on in lieu of class tomorrow. You also have been assigned a parasitic pathogen, which can also be worked on. The fungal brochure is due 3/28, so in 2 weeks from tomorrow. And then the I'd like the parasitic pathogen brochure due the first week in April, so 4/4. If you have any questions, shoot me an email or text. I'd be happy to address those concerns. So the other thing I want to draw your attention to is, again, this prokaryotic concept map, which has on these tabs below the GPC, the gram-positive cocci, a tab for the gram-negative cocci, and a tab for the gram-negative and gram-positive bacilli that you are going to want to understand the genres of and be able to reference this in, with relative efficiency when you're taking the exams or quizzes. So I wanted to draw your attention to that, and then also bring up the appendices that we've, looked over in class before. This appendices is available in DocShare. All these documents are available in the DocShare. And it covers, microbial agents of disease. We've really only talked about bacterial pathogens at this time and touched briefly on mycobacterial pathogens. So they're organized according to the organ system. These would be skin pathogens. And then we, over the next coming weeks, the second half of the course, we'll be talking about fungal, parasitic, and viral pathogens as well. So as you page through this appendices, you'll see that the next page is central nervous system pathogens, again, bacterial, fungal, parasitic, and viral. Cardiovascular pathogens, respiratory pathogens, urinary and reproductive pathogens, digestive pathogens. And then the Appendix B is the table of antimicrobials. So the first page of Appendix B is antibiotics or antibacterials. Antibiotics are only to treat bacterial infections, generally speaking. There's also antimycobacterials, which would treat your tuberculosis pathogen, or TB-like organisms like your Mycobacterium leprae. So each column is designated to a particular bacterial pathogen. The broad spectrums will be have a spectrum of activity that will cover both gram-positive and gram-negative. There are a few, like your metronidazole, which can also treat fungal, but we'll get into that later in Unit 3. Your gram-negative treatments would be this second column, the polymyxin B, imipenem and meropenem, piptazobacc, or chlorithral. Gram-positive treatments, vanco, linezolid, bacitracin, and rifampin, although I think rifampin, you can see here, also treats mycobacterium. So there's isoniazid, ethambutol, pyrazinamide, dapsone, and rifampin. I also have in parentheses some of the common names or the trade names that you would know. These are the generic names that we understand these antibiotics to be. So azithromycin is also commonly known as Z-Pak. Let's see, trimethoprim sulfamethoxazole oftentimes is referred to as Bactrim, or Septra would be some names that that's marketed under. And then in Unit 3 and 4, we'll be talking about these antibiotics, however, or antimicrobials. However, it's good for you to be familiar with these just because it's part of like Chapter 10, which is a chapter that's covered in Unit 2 somewhat. So I've narrowed and focused Chapter 10 down tremendously, but all of these antimicrobials are covered in exhaustive detail in Chapter 10. For Appendix C, I have a list of the different categories of microbes. So you have bacteria, mycobacteria, fungus, parasite, and virus. And then on each row is designated as a type of test. So in all of your presentations, we've covered how do you test for these different organisms. There's the direct smear method, which we use like the gram stain for bacteria, or the acid-fast stain for mycobacteria. But you can also do, and we will learn this throughout the remaining weeks in the course, there are some direct smears that we can use for other kinds of pathogens. The plate culture method is generally used for bacteria, mycobacteria, and even fungus. Not usually a good test method for parasites, though, as parasites need to be in a living system in order to be viable. And then cell culture for viruses. So this is also a table you'll want to be familiar with. And then I think what I would recommend then for studying is looking over these appendices, being able to quick reference things on them. When you look at your dichotomous key, be able to reference information on these relatively easily. And your presentations, I tried to make sure they covered the majority of genres that are mentioned in the dichotomous key and in the appendices, but it's important to be that you're accountable for all information on any of these references, from the screencast to these appendices to the dichotomous key to your brochures. You're held accountable for all of these things. There's 90% of it is overlap. But there are maybe some slight inconsistencies. For instance, the Virid and Strep group wasn't something that was presented. I'll cover it a little bit in the screencast, but it really you have to take some time. I don't believe Strep agalactiae was talked about in the presentations either, but I'll talk about that in the screencast today. So getting on with the screencast then, I'm going to cover gram-positive pathogens now, and then I'll move into a part 2 of this unit 2 screencast, which will be based on or focus on just gram-negative pathogens. So for gram stain morphologies, for bacterial morphologies, you have for single cells of bacteria the shape coccus, which is round, coccobacillus, which would be slightly elongated, not perfectly round. Bacillus would be elongated even more so. So the coccobacillus, although it doesn't look like it here, it's more ovular. True bacillus would be the length being considerably longer than the width. And then there's something called vibrio, where it's kind of club-shaped. Some bacterial cells can be club-shaped. We also have spirillum, which is kind of a spiral or helical sort of spiral or kind of squiggly sort of appearance. The spirochete would be corkscrew-shaped, and then pleomorphic means it has many shapes, actually. So you can see this looks like a wishbone, a doggy bone, and then this looks almost like a pin, a sewing pin. Arrangements for cocci, you can see sometimes on the gram stain this appearance of like a diplococci where it's in pairs. Streptococci would be chain of pearls. Tetrads, micrococcus tends to be in tetrads. So you may have seen on your lab competency, some of you gram stained the micrococcus, the very yellow colored colonies from the blood agar. That micrococcus tends to be in tetrads. Sarcinae, which is just a grape clustery kind of appearance, and then staphylococci. I really consider these to be pretty much the same. I haven't really seen any distinctive genres that are Sarcinae I really just consider this clusters. And really, even tetrads, if we see it on the gram stain, it has to be profoundly obvious for us to actually report tetrads. We would usually report it cocci either in chains or in clusters. Bacillus can be just single cells, or they can chain end to end, or they can do something called palisading, where they're side-side. And then we also have this V-shaped, sometimes we call this like a Chinese letter formation. There are certain organisms that'll, certain organisms that are bacilli appearing in kind of a Chinese letter fashion. So when we look at oops, sorry about that. I kind of lost my, there we go. When we look at gram-positive bacteria, your cocci are going to be the genres of Staphylococci, Streptococci, Enterococci, and Micrococcus, which is nice because it has coccus right in the name. The bacilli, there are gram-positive bacilli of the Bacillus genre, Clostridium genre, Listeria, Lactobacillus, and Corynebacterium genres. And this is all consistent with your dichotomous key. So we have some cool pictures. I think these are actually in your book. When it says SEM, that means a scanning electron micrograph. This is 2 micrometers. It looks like about an inch long. Would be considered 2 micrometers under an SEM. And you see this is like a cocci that are clustering. We also have bacillus. Now this is Bacillus thuringiensis, which is a lab-safe strain. And then they've got this Bt toxin, which is kind of, it looks like yellow filter paper, kind of worked in, which is cool. Staphylococcus can cause scalded skin, so this is a picture that I believe your book has of scalded skin. There's toxic shock syndrome, which can cause an impetigo. These are all staph-borne. So we've got, for staph diseases, your toxic shock, bacteremia, endocarditis would be a heart inflammation of the lining in the heart. There's pneumonia, and osteomyelitis would be a bone infection. Let's see, streptococci, we have for organizing streptococci something called the Lancefield classification system. So Lancefield groups A and B are what we've talked about the most in this class. A would be your strep pyogenes, strep pharyngitis, and then we also have group B, which is known to be like genital, or it can be skin pathogen of the skin. But group B is also known to pass the placenta and infect infants. We have for diagnosis, we can do a gram stain. We can do plate cultures also. It's not mentioned here, but you should know we do plate cultures. We also have some genetic tests in the case of a methicillin-resistant Staph aureus. There's also vancomycin, chloramphenicol, trimethoprim. I think these are all drugs that are listed on your appendices. And antisepsis, so good hand washing is important to keep from transmitting. For streptococci, you have strep A, group A strep, which is strep pyogenes. There's a list of illnesses here, which were all covered in your brochures, I believe. Erysipelas maybe wasn't mentioned, though. And you can see toxic shock can also be a strep pyogenes pathogen. Necrotizing fasciitis is a hallmark. Strep pyogenes infection, rheumatic fever is involving the valve in the heart. Glomerulonephritis is a kidney infection. And here are some pics of that. These are taken from your book, I believe. Group A strep, diagnosis is through gram stain. You can also do plate cultures. Treatment, pen is usually the front line, but if someone's allergic, they can do tetra, urethral, or clinda. Group B strep, as I mentioned, this is a neonatal bacteremia pathogen. Tends to cross that blood-brain, or tends to cross that placenta. There's meningitis, pneumonia. You can do, you can diagnose using an ELISA test, which is an immunotest. There's PEN-G, which would be the drug of choice for that. And I am going to have to probably wrap this screencast up. I will continue on with the gram-positive pathogens and also do the gram-negative pathogens in the next part 2. So peace out and stay tuned for part 2. Thanks.